Okay, everyone, so we will practice for the final exam. And the final exam will be over all the chapters that we learned, chapter 1 until chapter 17. So for best result of this exercise, please make sure you study chapters 1 through chapter 17 before taking this exercise. Prepare some pieces of papers and pen or pencil to write your answer. There will be 2 or 3 seconds after each question to write your answer on paper before the answer key is given and explained. You can always pause the video if you need more time. After finish the exercise, check your answer, how many are correct and how many are incorrect. Repeat the process until you get 100% correct answers. Make sure that the 100% correct answer are based on your knowledge, not from your guess. It is very likely that you will get a good score on your final exam if you follow this instruction correctly. Okay, we start with number 1. Which of the following is the first step of scientific methods? A, B, C, or D? Okay, remember, always start with observation. So start with the observations when we perform the scientific methods. Number 2, all organisms consist of cells. Because bacteria have cells, bacteria are an example of organisms. This is an example of blank reasoning. Inductive, constructive, productive, or deductive. Okay, so remember, this is based on general things, and then you make a conclusion from general things or knowledge. So this is a type of deductive reasoning. This is opposite of the scientific method. So scientific method usually start with the observations, and then tests, and then result, then you have the conclusion. That's inductive. So this one is deductive reasoning. Which of the following is the most inclusive taxon? Species, phylum, class, or domain? So remember, inclusive means the taxon that consists all the other taxon. So if we start from the top, the most inclusive one will be domain. Okay, there are 3 domains of organism. Remember, bacteria, archaea, and eukarya. And then under the domain, there is a kingdom. And there are 5 kingdoms of organism. Under the kingdom, there is a phylum. Under the phylum, there are classes. Okay, and then classes. Under the classes, there are orders, family. Okay, under the family, there is a genus or genera, and then finally species. Okay, so if you see this one, the most inclusive one will be domain. The most exclusive one will be the species. Okay, so the answer for this will be domain. Which of the following is the most inclusive level of organization? Atoms, cell, populations, or biosphere? Okay, from the organization, we start from the smallest one, the most exclusive one, which is atoms. Atoms will form molecule, and molecule become cell. Cell organized to make tissue. Tissue organized to make organs. Organs then making what we call the system or organ system. Organ system then become organism. Okay, and then organism become species, and species, they group together, the same species make a population. Populations will make the community, which is a group of living organisms, and community become ecosystem. All the ecosystem on the earth is called biosphere. Okay, so the most inclusive one in this case will be biosphere. Okay, number 5. Which of the following is the correct way of writing human species name in scientific journals? Okay, remember, it has two names. Okay, the first name start with capital letter. The second name start with lowercase. And both names have to be italicized. So from this rule, then we know C will be the correct answer. Both of the names are italicized. The first name start capital letter. The second name start with the lowercase. So C is the correct answer. Which of the following is the first part of format of writing a peer review paper? Always start with, of course, after the title, there will be abstract. So abstract will be number 1 things that you have to put when you write a paper in a journal. Which of the following property of life known as internal normal condition within a range that must be maintained continuously? A, B, C, or D? So this condition, internal condition, like our blood pressure, it has to be certain number, maybe between 115 to 120, over 80 to 70 to 90, for example. And our body temperatures around 95 to 98. That things, the internal body condition that has to be maintained is called the homeostasis. Okay, homeostasis. Based on the cell structure, bacteria and archaea are A, B, C, or D. Protist, animal, eukaryote, or prokaryote. Yeah, remember bacteria and archaea, they do not have nucleus. Okay, so all organisms that do not have nucleus is called prokaryote. Pro mean before, karyote mean nucleus. It means that no nucleus. Prokaryote for bacteria and archaea. The subkingdom of protists that has more plant characteristic is called protozoa, algae, bacteria, or archaea. Of course, bacteria, archaea is not protist. Okay, protist consists of protozoa and algae. Protozoa mean protists that look like zoo. Zoo mean animal. Okay, so that's not the answer. Okay, because the question is the protist that has more plant characteristic. This one should be the algae. Blank is a level of organization that consists of living organism and their environment. Living organism and their environment. Species, population, ecosystem, or community. Okay, species is only organism, one type of organism. It's not the answer. Population is a group of species, the same species, only living organism. Community also only living organism, which is a group of population. If we talk about the environment, then it will call as ecosystem. So ecosystem consists of living and non-living. Blank is level of organization that consists of living organism only within different species. Species, population, ecosystem, or community. Already explained that if it is organism only, living organism only, and different species, it will be community, because it is the same species is population. Okay, ecosystem with living and non-living. Okay, so the answer will be the community. In general, science follow the blank reasoning. Abstractive, constructive, inductive, or deductive. Okay, we mentioned this in number 1. Okay, so science, which is scientific methods, will follow the inductive reasoning, which is start from observation, experiment, and finally have the reasoning or conclusion from the experiment. And this type of reasoning is called inductive. Which one is not property of life? Growth, homeostasis, reproduction, or stay the same? Hey, of course, the one that is not property of life, when something stay the same, it is not living. Okay, so growth is property of life, homeostasis, of course, property of life, reproduction, every living thing should reproduce, so this is also property of life. So stay the same is the answer. Is the type of science that aims to use science to solve immediate problems. Applied science, basic science, natural science, or fundamental science. To solve problem, usually we use application. So they're going to be applied science. Which one is an example of applied science? Biology, chemistry, pharmacology, or physics? The answer will be pharmacology. Usually, when we learn pharmacology, we learn about medicine to solve problem, which is disease. So pharmacology more to the applied science. Which kingdom is considered as the first form of life on Earth? Monera or prokaryote, Animalia, Protista, or fungi? Of course, okay, the first form of life will be one cell organism, which is prokaryote, like bacteria. So the answer will be Monera or prokaryote. Which of the following is not one of the subatomic particles of an atom? Neutron, proton, electron, or nucleus? Okay, the answer will be nucleus. Okay, neutron, protons, electrons, they are subatomic particles. Which of the following subatomic particles are located on the shell of an atom? Nucleus, proton, neutron, or electron? Okay, if you see an atom, it has nucleus that consists of proton, and neutron. And then outside the nucleus, there is a shell. It's called the electron shell because it consists of electron. Okay, so electrons, the one that will be on the shell. So the answer will be electron. With atomic numbers of 6, okay, with atomic numbers of 6 and atomic mass of 15, the atom C will have blank. A, 6 protons, 6 neutrons, and 6 electrons, 9 protons, 9 neutrons, and 6 electrons. 15 protons, 6 neutrons, 9 electrons, 6 protons, 9 neutrons, and 6 electrons. Okay, just remember, first, the atomic numbers mean the number of protons. So proton, because it, okay, over there is 6, protons will be 6. And this is the rule that the number of proton will be equal to the number of electron. Therefore, the electron will also be 6. Okay, the atomic mass is the number of proton plus neutron. So 15, okay, equal proton plus neutron. Because we know that proton is 6, okay, then the neutron will be 15 minus 6, which is 9. So look for that one. 6 proton, 6 neutron. Oh no, 6 proton, 6 electron, and 9 neutron. Look for that one. 6 proton, 9 neutron, and 6 electron. So the answer will be D. Atomic number refers to the number of, I just mentioned that, number of proton. So D is the answer. Okay, now from number 21 until 30, this is a true and false. Okay, 21. Atomic mass refers to the number of proton and neutron. Yes, that's correct. Proton plus neutron will tell you the atomic mass. So this one will be true. Okay, number 22. In an atom, the number of proton is equal to the number of neutron. No, okay, it's not always. Protons will always equal to electron. So this one should be false. 23. Atom that loses and gains protons are called elements. No, it's not true. It is called ions. So this is another false. Okay. 24. Positive ions are called anions. No. Positive ion is called cation. So this is false. Should be cations. 25. Type of chemical bonds where the atoms share their electron is called hydrogen bond. Another incorrect statement. Okay. So chemical bond, the type of that share electron is called covalent bond. So this is another false. It should be covalent bond. All chemical reaction in an organism is called catabolisms. Okay, another incorrect statement. It should be metabolism, not catabolism. Next, the synthesis reaction are known as anabolism. Yes, remember ana mean synthesis, kata mean breakdown. So ana is the builder, cat is the destroyer. Okay, so that's true. Photosynthesis is an example of anabolism. Yep, the name is telling you this is synthesis. It should be anabolism. That's correct. Octet rule mentioned that there are maximum of 8 electrons can be present on the second electron shell of an atom. Yes, 8 mean octet, and it is from the second electron shell until the next one. Okay, so the outside should be 8, except the first electron shell will be only 2. It's called the duplet rule. Last one. Covalent bond is the weakest type of chemical bond. No, okay, covalent bond is the strongest type of chemical bond. The weakest one is called the hydrogen bond. So it should be false. Okay, it should be the strongest type of chemical bonds. Okay, for number 31 to number 40, you have to fill out the blank. You have to type your answer over here. Number 31, blank are substances produced at the end of reaction. Okay, remember, reaction consists of reactant, like for example, A plus B become C. So A and B is called reactant. C is called product. Okay, so the substance that produced is called product. Blank is solution that dissociate into cation and hydroxide ions. Okay, what is that? So whenever you produce hydroxide ion over here, it means it is an alkaline or base. See? So this is a alkaline or base. Solution that has pH lower than 7 is called blank solution. Lower than 7 is acid. Okay, it has to be acidic solution. Blank is the most abundant element of life, which account for about 65% of all elements of life. So which element is that? So remember, our body only consists of 6 major elements. The rest will be minor elements. So oxygen, carbon, hydrogen, nitrogen, phosphorus, and sulfurous. Okay, the most abundant one is the oxygen, 65%. Blank is anything that takes space and has mass. It is matter. So matter is anything that takes space and has mass. Macromolecule consists of individual subunit is called. Okay, so macromolecules, the polymer, it will have subunit is called the monomers. Okay, so macromolecule like proteins, carbohydrates, lipids, nucleic acid, they are polymers. So they will have what we call the monomers as their units. Blank is the storage molecule of carbohydrates or polysaccharide in animal or human. Okay. If it is in the plant, it's called starch. In the animal, it's called the glycogen. Type of protein that catalyze reaction inside an organism are called catalyze. Catalyst will be enzyme. So enzymes are catalyzing reaction. Blank is a process that changes protein structure and function. It is what we call the denaturation. Okay, so denaturation protein means that the protein is actually the structure will be changed or broken down. Blank is the diverse group of non-polar, so non-polar molecules or hydrophobic molecules. So all non-polar molecule is called lipids or fats. Okay. Okay. This is the next group, 41 until 50, which is matching. Okay. So you have questions on the left and answer on the right side. We start number 41. The building block or monomer of carbohydrates. Carbohydrates is also known as polysaccharide. Therefore, the monomer is called monosaccharide. Okay, so E will be the answer for that one, monosaccharide. The building block or monomer of proteins. Proteins consist of many amino acids. Okay, so the monomer of protein is called amino acid. The building block or monomer of lipids. Lipid consists of fatty acids. So look for the fatty acids over here. The building block or monomers of nucleic acid. Nucleic acid like DNA, RNA, they consist of nucleotides. So the monomer is called nucleotides. An example of primary structure of proteins. So primary structure is the structure of protein that consists only a sequence of amino acids. Example is insulin, okay, bovine serum insulin only have primary structure. An example of tertiary structure of proteins. Enzymes are example for this one. Enzyme has a ball-like structure, which is 3D structure, and it is a tertiary structure of protein. So it is enzyme. An example of quaternary structure of proteins. So the quaternary structure of protein is actually consists of several tertiary structure. Example is hemoglobin. Hemoglobin actually consists of 4 tertiary structure that combine together. Okay, so it's called the quaternary structure of proteins. The type of reaction that break a polymer into monomers. So if we want to break polymer into many monomer, we just put water on it. It's called the hydrolysis. So the water will split bigger molecule into many smaller molecules. The type of reaction that makes polymer from monomers, this is going to be opposite of hydrolysis. It means that we have to take water out. It's called the dehydration. So the process is called the dehydration synthesis. One of the control dogma of life. Okay, start from DNA into RNA, and then RNA into proteins. DNA to the RNA is called the transcription. RNA to protein is called the translation. So this is the only choice that we have over here, which is the translation of RNA into protein. So translation. Okay, next is another multiple choices. The smallest structural and functional unit of life is going to be cell. Life or living thing consists of cell. So cell will be the smallest unit of life. Which of the following organism is prokaryote? Again, prokaryote means no nucleus. Which one is that? Algae is not. Yes, bacteria will be the answer. That's should be the answer right away. Which of the following organism does not have ribosome? Bacteria, protozoa, animal, plant, or none of the above? Remember, all cell, it doesn't matter prokaryote or eukaryote, they all have ribosome. They all have chromosome. They all have cell membrane. They all have cytoplasm. Okay, so the answer should be none of the above. All cell will have ribosome. Which of the following organism is an autotroph or producer? Which one? Bacteria, protozoa, animal, plant, or fungi? So autotroph means organism that able to make food through the photosynthesis. This organism will have chloroplast, which has a green color. So look for green color over here. Bacteria, protozoa, animal, plant will be the answer. Okay, so plant and algae, they have green color because they're able to perform photosynthesis. So they are autotroph or producer. The main framework of cell membrane is the protein bilayer, nucleic acid bilayer, cholesterol bilayer, or phospholipid bilayers. So the cell membrane consists of two layers of phospholipid. Okay, so phospholipid is molecule that have head and two tail. Okay, so the answer will be phospholipid bilayers. The structure of protein that span the cell membrane is termed as A or N, integral proteins, peripheral proteins, linker, or receptor. So remember, cell membrane, it is bilayer of phospholipid. Okay. They also have proteins. Okay, if that proteins span between outside to inside, that one is called the integral. If it is only outside or only inside, this is called as peripheral protein. Okay, so linker mostly only the outside, so they are peripheral. Receptor also mostly outside. Okay, that also peripheral protein. So the answer will be integral proteins like ion channel or transporter. What is a hypertonic solution? A solution that has higher concentration of impermeable solutes than a cell, a solution that causes cell to burst, a solution that has the same concentrations, or the cell have lower concentrations. Hyper mean higher. Okay, it should be a solution that have higher concentration of impermeable solutes compared to the cell. So that should be the answer. What will happen if a cell is placed in the hypotonic solution? Water will enter the cell, causing crenation. Water will leave the cell, causing crenation. Water will enter the cell, causing hemolysis, or water will leave the cell, causing hemolysis. So if it is hypotonic solution, it means that that solutions will have lower concentration compared to the cell. So let's say the cell have 0.9% and hypotonic solution don't have anything, so 0%. So what happened? The water will enter into the cell and the cell become bigger. And this one is called hemolysis. So water will enter the cell causing hemolysis. Enter to the cell, hemolysis. What will happen if a cell is placed in a hypertonic solution? That's going to be opposite than hypotonic solution. So the cell have 0.9, but the outside in the container, for example, it has 9%, okay, more solute concentration. Then what happened? The water will leave the cell and the cell become smaller. That condition is called the crenation. So leave the cell, causing crenation. The process of engulfing or eating solid particle or foreign cell by the white blood cell is called hemolysis, photosynthesis, phagocytosis, or pinocytosis. So, eating, phago means eating. So that should be the answer. So the process of eating molecule by white blood cell, usually like bacteria, will be eaten by white blood cell, and that process is called the phagocytosis, cell eater. Movement of oxygen enter the cell and carbon dioxide exit the cell are example of simple diffusion, facilitated diffusion, osmosis, or active transport. Okay, remember oxygen and carbon dioxide, they are very small molecules, hydrophobic. So the oxygen will be enter into the cell easily, and the carbon dioxide will be exit the cell easily. And this one is called the simple diffusion without any help. They just cross the cell membrane easily. Which of the following reaction is catabolism? Breakdown of glucose in the cell to produce ATP, synthesis of proteins in the cell, replication of DNA in the nucleus, synthesis glucosis by the green plant. Okay, just remember cat or catabolism mean breakdown. Okay, the opposite is anabolism. Okay, anabolism means synthesis. Okay, look at the answer. Synthesis, B, it should be wrong because it should be anabolism. Replication is also synthesis, making more. So it is wrong. Synthesis glucose, again, this is a synthesis. This one is wrong. So the answer will be breakdown of glucose to produce ATP. Which of the following reaction is anabolism? Breakdown glucose, dehydration synthesis, breakdown of glycogen, or hydrolysis of proteins. Okay, so remember anabolism means synthesis. Look from these choices, which one is synthesis? Of course, A is not correct because it is breakdown. Dehydration, let's pass that one first. C, breakdown is not correct. Hydrolysis is also mean cutting bigger molecule into smaller molecule with the water. So this is also breakdown molecule. Okay, so the correct answer will be the dehydration synthesis. Which of the following reaction release energy? Metabolism, catabolism, anabolism, or endergonic reaction? Okay, remember the rule. If it is catabolism, Okay, it will break down bigger molecule into smaller molecule, and they always release energy. So when the reaction release energy, it's called exit. Okay, exergonic. Okay, so it should be breakdown reaction, which is catabolism. So that should be the answer. Anabolisms will be opposite, which is anabolism will require energy. Molecule that function to increase rate of chemical reaction is called, so increase chemical reaction, hormones, enzyme, substrate, or product. Substrate and product is not the answer, okay? Because this is actually the molecules that will be reacted, okay? Hormone is not for making reaction faster, so the answer will be the enzyme. A coenzyme is blank. Examples are blank. A, inorganic molecules, zinc, iron, copper. Organic molecules, like vitamins. Inorganic molecules, like iron, copper. Or organic molecule that denatures proteins, like vitamins. So enzyme, so it should be reaction that, molecule that increase reaction. Okay, so it should be enzyme. Okay, and that, if it is coenzyme, that will be organic molecules. An example of coenzymes are vitamin. So vitamin actually organic molecules, part of an enzyme. What term is defined as the ability to do work? Energy, metabolism, oxidation, or reduction. So this is the definition for energy. So energy is defined as the ability to do work. A hormone or secretion that affecting nearby cell or neighbor, okay, neighboring cell is called paracrine, endocrine, exocrine, or autocrine. So if it is distant cell, is endocrine. So the cell are far away. If it is itself, is called the autocrine. Okay? If it is neighbor, it's called para. So the answer will be paracrine, the hormones that will affect the neighboring cell. How does lipid-soluble hormone affect change in a target cell? So lipid-soluble hormone, by directly stimulating protein synthesis or altering the gene expression, promoting phagocytosis, causing synthesis of second messenger, stimulating protein kinase in the cytoplasm. Okay, remember lipid-soluble hormone, it will enter into the nucleus. And the receptor for this lipid-soluble hormone will be in the gene, in the DNA. So it means that, okay, the effect will be altering the gene. So it will be A, okay, altering the gene expression. How does lipid-soluble hormone behavior on the target cell membrane? Diffuse easily, cannot diffuse, require channel, or stay outside. Okay, so lipid solubles will be entering the cell easily. Therefore, their receptor will be inside the cell, which is inside the nucleus. So it's going to be diffuse easily into the cell. Okay, for number 71 to 80, this is true and false. 71, the last stage of Calvin cycle is the carbon dioxide reduction. True or false? It's going to be false because it should be the RUBV regeneration. 72, a channel protein used by water to enter and exit the cell is called aquaporins. Yes, it is true. Movement of glucose enter the cell using transporter is an example of simple diffusion. It is not. It is called a facilitated diffusion because it's require facilitation, which is transporter. Catabolism is an exergonic reaction. Yes, we just mentioned that. Every catabolism will release energy, and it is called the exergonic energy exit. Okay, exergonic reaction. Competitive inhibition slows down reaction without possibility to reach the maximum speed. It is incorrect. It will slow down, but it will reach the maximum speed when we increase the enzyme or the substrate. All chemical reactions in an organism are called metabolism. Yes. Okay, so all chemical reaction is called the metabolism that consists of anabolism and catabolism. The synthesis reaction are known as catabolism. Nope, it should be anabolism for synthesis. Catabolism is for breakdown or decomposition. Photosynthesis is an example of catabolism. Again, it is incorrect. If it is synthesis, it has to be anabolism. Autocrine is a type of hormone that acts to a distant target cell. No, auto means self, okay? So it should be for itself. So the target is actually itself or its own cell. A lipid-soluble hormone has second messenger to alter the process inside the cell. No, the second messenger only for water-soluble hormones. Lipid-soluble hormone will not have the second messenger. Okay. Number 81 to 90 will be fill in the blank. Okay. 81. Blank is the light spectrum color that is not absorbed by chlorophylls. What is the answer? Yes, the answer should be green. The reason why we see leaf green because the green color is reflected to our eyes. It is not absorbed by the leaf, by the chlorophyll. Blank is the type of electron microscope that can show 3D image. Yes, it should be scanning electron microscope or SEM. The plant and algae cell wall are made of blank. Plant and algae or algae cell wall is made of cellulose, which is very rigid type of carbohydrate. It's difficult to be digested in our stomach. Blank is the molecule that strengthens the cell membranes. It should be cholesterol. So we need cholesterol to make our cell membrane become stronger. Type of membrane protein that receives signal such as a hormone is called, okay, is a receiver, okay, receive signal. It is called the receptor. Okay, receptor. Blank is the type of hormone that act upon distant target cell. So if it is distant target cell, it's called the endocrine or endocrine. Blank is the first step of cellular respiration that is occurring in the cytoplasm. Right? Cellular respiration start with glycolysis. It is happening in the cytoplasm. Blank is the first step of photosynthesis that is happening on the thylakoid membranes. That process is called the light reaction because it requires light or photoreaction. Blank is the molecule that enters the Calvin cycle of photosynthesis. So this one will be carbon dioxide. So carbon dioxide will enter into the Calvin cycle. It will be then converted into carbohydrates. Last one. Blank is the molecule that enters citric acid cycle or CAC of cellular respiration. Okay, the molecule that enters citric acid cycle is called the acetyl-CoA, acetyl coenzyme A, which is the product of pyruvates conversion. Okay, the next one is matching. So you have question on the left and answer on the right side. Okay, 91. The control center of the cell. So this is talking about the organelles, the name and the functions. So the one that control the cell or control center of the cell is the nucleus. So look for the nucleus, which is this one, I. The one that produces proteins is going to be ribosome. Organelle that produce lipids and carbohydrates, it will be smooth ER. The organelle that modify proteins and lipids to make the final products, like enzyme and hormone, it should be the Golgi apparatus. Okay, the organelle that carry enzyme that destroy foreign cell or foreign molecules, okay, it should be lysosome. The organelle that carry enzymes that detoxify toxicants or drugs, it should be peroxisome. The organelle that carry enzyme that destroy unneeded proteins, and the name is telling you, it's going to be proteasome. The organelle that produce ATP, so this is your source of energy, your power plants inside the cell, which is the mitochondria. The organelle that produces foods or glucose. So these ones will be in the plants only, with organelle that has green color, which is the chloroplast. Last one, control movement of molecules in and out the cell. So part of the cell that control movement of molecule in and out of the cell will be the cell membrane. Okay, so next one, another multiple choices. Which of the following is not the function of mitosis in multicellular organism? Reproduction, growth, repair, or replacement. Remember, for multicellular organism like human, mitosis is used for growth, repair, replacement. It will not use for reproduction because reproduction will use the meiosis. So that should be the answer. Which of the following organism has diploid dominant life cycle? Animals, bacteria, plants, or fungi? Diploid dominant like human. So we are actually diploid because our cell consists of pairs of chromosome, which is double or diploid. So it will be animal. In human, the haploid cell carry blank chromosome, 11, 23, 46, 92, or 184. So remember, human diploid cell, okay, most of the cell in human will be diploid, will have 46 chromosome. Okay, if the cell is haploid like sperm and the eggs, then only carry half of the diploid, which is 46 divided by 2 will be 23. So 23 chromosomes. Which of the following cells are haploids? Skin cell, brain cell, sperm, bone cell, or spermatocytes? Okay, remember in humans, the haploid cell will be sperm. It is for male, or eggs if it is for female. So look for this, whether sperm or eggs. So it should be C, which is sperms. The reproduction of bacteria is called the sexual reproduction, binary fission, doubling time, fertilization, or meiosis. So bacterial reproduction is called binary fission because that bacteria will always reproduce from 1 into 2, 2 into 4. So every cell will become 2. That's what we call the binary fissions. The time needed by bacteria to double in number is called sexual reproduction, binary fission, double or doubling time, fertilization, or meiosis. Of course, because this is double, then the time is called the doubling time. Okay, now if a doubling time of bacteria is 20 minutes, okay, how many bacteria will be after 1 hour? So 1 hour is equal to 60 minutes, right? Okay, so it is said that bacteria has 20 minutes double time. So every 20 minutes, the bacteria become double. So how many will be after 60 minutes? Okay, so from 1, 2, 4, 8. Okay, this is 20 minutes, 20 minutes, another 20 minutes. It's going to be 8. Okay, so after 1 hour, the bacteria become 8. Which of the following stages is the longest stage during cell life cycle? Prophase, metaphase, anaphase, telophase, or interphase? Remember, life cycle, okay, consists of interphase, which is the longest part, okay, and the interphase itself consists of G1, S, and G2. And the mitotic phase, that consists of prophase, metaphase, anaphase, and telophase. So the longest stage will be interphase. Which of the following stages is the chromatin presence? Okay, which of the following states when the chromatins are present? Prophase, metaphase, anaphase, telophase, or interphase. So when cell start to divide, okay, perform the mitosis, so the interphase, okay, during the interphase, we will not be able to see the chromosome. It's just thin thread is called the chromatin. Okay, when they start the prophase of the metaphase, the chromatin become chromosome. Okay, so therefore chromatin is only present during interphase. Which of the following stages is the nuclear envelope disappear or breakdown? Prophase, metaphase, anaphase, telophase, or interphase. So when the cells start to perform mitosis, it start with the prophase, and during this time, the nuclear envelope will be broken down, okay, and the chromosomes start to divide. So you're gonna see X structure, right? And the spindle fiber will start to forms. Okay, that's all happening during prophase. Which of the following states does the nuclear envelope reappear or reform? So start from prophase, the nuclear envelope breakdown, and then metaphase, the chromosomes will be lining in the middle. Okay. Anaphase, the cell elongate, the chromosomes will be separate into the two poles. Okay, so the chromosome will be double. And then telophase, this is when the cleavage furrow present and the nuclear envelope reform. Okay, so this is telophase. Nuclear envelope reappear or reform will be during telophase. Which of the following states does the cell elongate? I just mentioned that, I just explained before that. Okay, it's gonna be the anaphase, during anaphase. Which of the following stages do the chromosome align in the center or the middle of the cell? Okay, so this one will be during metaphase. Which of the following stages does the cleavage furrow present? Okay, so this one during telophase. This is cleavage furrow over here. Okay, nuclear envelope reform. Okay, it will be telophase. How many chromosomes are there in every diploid human cell? Of course, every human cell except sperm and eggs will have 46 chromosomes. Okay? 43 chromosomes or 23 pairs of chromosomes. Okay, so 46. During mitosis of human cell, there are blank chromosome at prophase. Okay, prophase is when the chromosome start to divide, but it's not dividing yet. Therefore, the number of chromosome will be the same, which is 46. During mitosis of human cell, there are blank chromosome at metaphase. Metaphase, when the chromosomes lining in the middle, all of them, and still will be 46. Okay, it's not divided yet. So still 46 during metaphase. Okay, during mitosis of human cell, there are blank chromosome at anaphase. Okay, remember anaphase when the cell elongate and the chromosome already divide, they're gonna move to the opposite pole. So every pole will have 46. This is 46. So total chromosome now is double, become 92. During mitosis of human cell, there are blank chromosome in telophase. So telophase is when the cell start to divide, and there is a cleavage furrow, the nuclear envelope reform, the chromosome will be inside the nucleus. It's going to be 46 on the top, 46 at the bottom, but the cell is not divided yet. So the number of chromosomes within the cell will be 46 plus 46, which is 92. After mitosis of human cell, there are blank chromosomes in each daughter cell. So after telophase, like this, okay, with 46, 46 on the top, 46 on the bottom, then when the cell divided into two daughter cells, then every daughter cell will carry 46 chromosomes. Okay, for number 121 to 130, this will be true and false. 121. There will be 92 chromatids present at telophase stage of mitosis of human cell. True or false? It's gonna be false, remember, in the telophase, the chromatids will be no more. No more chromatids, okay? Because chromatids only present at prophase and metaphase. When it is divided, its chromatids become chromosome, which is in the anaphase and telophase. So telophase will not have chromatids. It is false. There will be 92 chromatids present at anaphase stage of mitosis of human cell. Again, it is another incorrect statement because during anaphase, the chromatids already separated into chromosomes. So there will be no more chromatids in anaphase or during anaphase. There will be 92 chromatids present at prophase 1 stage of meiosis of human cell. Okay, remember during prophase 1, this is meiosis. Okay, the chromosomes start to divide, but it's not divided yet. Okay, so each chromosome will have 2 chromatids. So because we have 46 chromosomes, then the number of chromatids will be 92 during prophase and also metaphase. So this one should be correct. True. There will be 23 tetrads present at prophase 1 stage of meiosis of human cell. So during prophase 1, okay, the chromosome, okay, each chromosome has 2 chromatids, and then 2 chromosomes, they will be joined together to make what we call the tetrads, okay, and the tetrad will have 2 chromosomes with 4 chromatids, okay, and because there are 46 chromosomes, then if every 2 will combine, then become tetrad, then you only have half, okay, number of tetrads compared to the number of chromosomes. So human have 46, then the tetrad will be 23. So that should be correct. Each human cell carries one circular double-stranded DNA. True or false? It is not. Okay. Human cell carry 46 chromosomes. It's not only one. So it's carry several double-stranded DNA. In addition to its chromosome, many bacteria carry small circular double-stranded DNA. Is called plasmid. That should be true. Okay. During mitosis in plant cell, cleavage furrow will form at telophase stage. Okay, remember it is plant cell. Plant cell do not form the cleavage furrow. Okay, they create what we call the cell plates. It's not cleavage furrow, so it should be incorrect. The part of chromosome that holds sister chromatids is called kinetochore, which is not correct. Okay, the one that holds the sister chromatids together is called the centromere. Chiasma or chiasma is a part of tetrad that hold sister chromatid together. It is incorrect because the chiasma or chiasma will hold the non-sister chromatid together. Crossing over is present during prophase 1 and prophase 2 of meiosis. It is incorrect because crossing over only present during prophase 1. Okay, for 131 to 140, it's going to be fill in the blank. Blank is the type of checkpoint during cell cycle that determines whether conditions are favorable for cell division or not. Okay, what checkpoint is that? Okay, you have G1 checkpoint, you have G2 and M checkpoint. So this one should be the G1 checkpoint. Blank is an example of positive regulator during cell cycle that promotes cell division. Positive regulator that promotes cell division is called a proto-oncogene. If there are mutations of positive and negative regulator of cell cycle, the cell could undergo uncontrolled cell division, commonly called as cancer or tumor. The final product of mitosis are blank. So what are the final products? Will be two identical daughter cells. Each spermatocyte will produce blank as the final product. Remember, this is each spermatocyte. One spermatocyte will produce four sperm as a final products. In a normal and healthy males, there are blank produced per day. Okay, so healthy male will have million of spermatocytes. Every spermatocyte produce 4 sperm. Okay, so a male actually can produce about 300 million sperms per day. In a normal and healthy female, there are blank produced per month or period. Okay, so for female, they actually only produce 1 eggs per month or per period. The process of unification of sperm and egg is called blank fertilization. karyokinesis is famously known as karyokinesis, which is nuclear cell division. It is famous as mitosis. The plasmid and the foreign gene inserted to it is called blank that will produce a recombinant proteins. Okay, so the plasmid and the foreign gene is called the recombinant DNA. So the foreign cell DNA will be combined with the plasmid of bacteria, and they are called as the recombinant DNA. Okay, now the matching. Okay, so you have question on the left and answer on the right side. 141, the cell has 23 tetrads. Chiasma and crossing over are present. Okay, that should be, look at the crossing over here. It is only happening during the prophase number 1. So that one, prophase number 1. The cell has 23 tetrads and are aligning in the center of the cell. So this one will be metaphase 1. Okay, the cell has 23 chromosomes. No, this is chromosome, not tetrads, okay? That are aligning in the center of the cell. So if it is 23 chromosome, that stage will be metaphase 2. Okay? The 23 tetrads are separated into 46 chromosomes with 2 chromatids each. So this is separations is happening during anaphase. And because this is from tetrad, then it's going to be anaphase number 1. Each cell has 23 chromosomes that are separated. Its chromatids become a chromosome. Total, there are 46 chromosomes in each cell. So this is chromosome, okay, not chromatid, not tetrads anymore. So there will be anaphase number 2. Okay? So the chromosome is separated. Anaphase 2. Okay, chromatids are present. This is, of course, interphase. The nuclear envelope reappear. It is only in telophase, and because this is meiosis, this one reappear only during the telophase number 2. The cell separates into 2 cells. Each cell carries 23 chromosomes without nucleus. So it's going to be cytokinesis, but because there is no nucleus, there will be cytokinesis number 1. Each cell separates into 2 cells. Okay, each cell, total 4 cells, carries 23 chromosomes protected in the nucleus. So the cell separated, so it should be cytokinesis. And because each cell now carry 23 or haploids, that will be the product of cytokinesis 2. Cleavage fluorescent. Each pole has 23 chromosomes without nuclear envelope. Okay, so the cleavage fluorescent is mean this is telophase, but look over here, there are nuclear envelope reform. Then this will be telophase, but number 1. Telophase 1. (151) Which of the following is not one of many historical misconceptions about heredity? Sperm content fully formed of little humans. This is one of the misconceptions. Female control the trait, one of them. Genotype are expressed in phenotype. So this is not one of them. So this should be the answer. Place in the DNA strand that encodes information for a specific trait is called gene locus, messenger RNA, phenotype. Should be not. This is not DNA. This is a process of crossing. So the answer should be the gene locus. The alternative version of a gene is called a or an genotype. Oh, this should be phenotype, alleles, or genome. Alternative version of gene is called the allele. Okay, this should be maybe dominant or recessive allele. So genotype always consists of two alleles. The specific combination of gene carried by an individual, combination of gene that cause the phenotype is called genotype. Phenotype, of course, is not allele or genome. Okay, so genotype will be expressed into phenotype. So this one should be the answer. The characteristic version of trait that can be observed and measured. So something that you can observe and measure is called genotype, phenotype, allele, or genome. So part of the trait or version of the trait that can be observed is called phenotype. What is the probability of children for being albino if the mother is albino and the father is homozygous dominant? 0%, 25%, 50%, 75%, or 100%. Okay, remember, albino is a recessive abnormality. So person that have both recessive allele will have the albino. So the mother is albino. So we know the mother will have lowercase letters. Okay. The father is homozygous dominant. So the father will be heterozygous dominant, all capital A like this. Okay. So the children 100% will be heterozygous like this, which is normal or carrier. Okay, so the children actually have a normal skin color. So what's the question? What is the probability of children for being albino? So none of them. So it will be 0%. What is the probability of children for having polycystic kidney disease if the mother is normal and the father is heterozygous? 0%, 25%, 50%, 75%, or 100%. Okay, which one is your answer? Okay, remember PKD, polycystic kidney disease, is a dominant autosomal disease. So the one that have dominant alleles or homozygous dominant and also heterozygous will have the disease. So it is said that the father is heterozygous, which is the father has the disease, the mother is normal. So let's say we're using the same letter, okay, letter A. So for normal will be lowercase. And the father is actually have the disease, but heterozygous, like this. Okay, now put them in the Punnett square. Okay, so mother, you put it here. Father, you can put it here. Okay, this is the sperm. And then you combine for the children. It's going to be heterozygous, which is having the disease. This is normal, heterozygous, normal. So 50% normal, and the other 50% will have the disease. So it's going to be 50% of the children. What is the probability of children of having wavy hair type if both mother and father are curly? 0%, 25%, 50%, 75%, or 100%. Okay, so wavy hair, this is a type of incomplete dominant. Okay, so when the in the heterozygous, the characteristic will be in the middles. Okay, it's combination between the dominant and recessive. So look at this, mother and father are curly. So we can use C letter, capital C for curly. Okay, and the father is gonna be the same, capital C. Okay, so all the children will be curly. Okay, so this is curly hair, curly hair. Yeah, so the answer will be how many wavy hair? It's going to be 0%. None of the children will have wavy hair. What is the probability of the son for having color blindness if the mother is carrier and the father is normal? 0%, 25%, 50%, 75%, or 100%. Okay, remember color blindness is carried by the X chromosome. So it's only the gene only present in the X chromosome. Okay, we can use B letter, okay, for color blind. B or capital B or lowercase b. Now look at this, mother is carrier. Remember, mother will have 2 chromosome X. It is a carrier, so one of them will carry capital letter and the other one will be lowercase. Okay, this is carrier, which is actually normal. It is not color blindness. And the father is normal. So the father only carry 1 X chromosome and 1 Y chromosome. So because it is normal, we can put the capital B on the X. Okay, so the father is normal. Now look at the children. Again, we're going to put in the Punnett square. So this is for mother. So it's going to be X with B capital letter and then X carry lowercase. And father, X carry B capital letter. which is normal. And Y, because Y don't have the color blindness gene, so it does not carry that. Okay, so we combine this. This one will be girls. Okay, so daughter. The daughter will be normal on the top. This one, daughter, which is carrier. Okay, and this is gonna be son, which is normal. And this one will be son that has color blindness, because carry the lowercase b. Okay, so the son 50% color blindness and 50% normal. Okay, so this one normal, this one color blindness. Okay, so the answer will be 50% of the sons. The color blindness gene is located in chromosome number 1, number 21, X chromosome or Y chromosome. Okay, we just explained that the color blindness gene is carried in the X chromosome. The individual number or set chromosome and appearance. So this is definition for karyokinesis, karyotyping, karyotype, or karyogram. Kinesis is process, so this is not the answer. Karyotyping also process, it is not the answer. Okay, karyogram is photograph. The set will be this, the type, the karyotype. The photograph of individual numbers or set chromosome and appearance, karyokinesis, karyotyping, karyotype, or karyogram. Okay, I just explained that on the previous questions. The photograph is the karyogram. The method of knowing or determining the individual numbers or sets, chromosome, and appearance. Karyokinesis, karyotyping, karyotype, or karyogram. So this is the method. It means the process may be one of this. Okay, and this one is not. Okay, so to know your karyotype, you have to perform karyotyping. Just like if you want to know your blood type, you do the blood typing. So this should be the answer. The normal set and appearance of the chromosome is called, okay, it is normal. What it is called? Euploid, aneuploid, polyploidy, or tetraploid. Normals mean euploid. Normal set. So in human, 46 or 23 pairs. Which of the following chromosome set is an example of euploid individual, normal individual sets of chromosome? 46XX, 47XXY, 47 plus 21, or 45X0. Okay, of course, it is normal. For human, it should be 46. So that will be the answer, whether 46XX or 46XY for the male. So this should be the answer. Which of the following chromosome set is an example of monosomy individual? 46XX, 47XXY, 47 plus 21, or 45X0. So monosomy means it's less than normal. So normally 46, then if it is less, will be 45. Okay? So this is the answer. Which of the following chromosome sets is an example of trisomy individual? 46XX, 46XY, 47 plus 21, or 45X0. So trisomy means there are one extra chromosomes in the individual. So normally 46, this is normal. So if it is more than one, more than normal, then it's going to be 47 plus 21. So he has extra chromosome in chromosome number 21. Which of the following chromosome sets belong to Down syndrome individual? 46XX, 47XXY, 47 plus 21, 45X0. So Down syndrome is actually, it has extra chromosomes in pairs number 21. So this should be the set of chromosome for individual with Down syndrome, 47 plus 21. Which of the following chromosome sets belong to Klinefelter syndrome individual? 46XX, 47XXY, 47 plus 21, 45X0. So Klinefelter is actually trisomy. It has extra chromosome, which is extra X chromosome. Okay? So normally XX or XY, but this one have additional X over here. So this is 47XXY for Klinefelter syndrome. Which of the following chromosome belongs to the Turner syndrome individual? 46XX, 47XXY, 47 plus 1, 45X0. This is normal. This is Klinefelter. This is Down syndrome. So Turner is actually monosomy. It has less one chromosome, okay, which is losing one chromosome over here. So X0, 45X0 for Turner syndrome. Okay, 171 to 180, this is true and false. Human height and skin color are example of polygenic traits. True or false? Yes, it is true. So human height and skin color are caused by several genes. It's not only one genes or one pairs of gene, but it has several genes. So it is true. The first law of inheritance is the law of independent assortment. True or false? It is false. Okay, remember the first law is segregation. Okay, law of segregation. The phenotype ratios of F2 generations of dihybrid crossing will be 9, 3, 3, 1. So F2 for dihybrid is gonna be 9, 3, 3, 1. That's correct. The phenotype ratio for F2 generation for monohybrids, okay, so will be is only 2 observed phenotype, okay. It's gonna be 3 to 1. Yes, it is correct. Dominant allele is the allele that will be masked by the recessive allele. True or false? Is wrong. Okay, this is not the one that is masked, but this is gonna be the one that actually cover the other allele. Okay, it will mask. It will mask the recessive allele. When both alleles are dominant alleles, the genotype will be homozygous dominance. So both of dominant, homo, then it will be correct, homozygous dominant. The chromosomal theory of inheritance states that chromosome carry the gene. Chromosomal theory, chromosome carry the gene. That should be true. Mendel was the scientist that developed the chromosomal theory of inheritance. True or false? No, it is wrong. It is not Mendel. Okay, so this is false. The one that developed this theory is or are Sutton and Boveri. Non-disjunction is the ability to separate the chromosome during meiosis. True or false? It is false. Okay, it is actually not the ability, okay, but the inability or inability or disability or unable to separate chromosome. This is non-disjunction. Cry to cat is an example of abnormality of chromosomes, deletion of chromosome number 6, true or false. This is not number 6. This should be number 5, chromosome number 5. Okay, 181 to 190. This is filling the blank, so you have to write down your answer. Okay, number 181. Blank is a characteristic that individual has such hair type, hairstyle, eye color, skin color, and so on. Okay, what is this? This is what we call the trait. Okay, so traits will have the versions. Yeah, it's called the phenotype. Like for example, if it is hair type, the trait is hair type. Maybe the phenotype will be curly, wavy, or straight hair. Okay, the eye color, this is the trait. The phenotype may be blue, green, brown, or black. Okay. Blank is the type of crossing that look only for one trait of inheritance. So this is going to be mono. Okay, so it's going to be mono hybrid cross. The allele that masks another allele is called, the allele that cover another allele is called, it's going to be the dominant. All right, this is wrong answer over here. So this one should be dominant allele. Okay, so this is a wrong key. Okay, let's see what happens next. If the heterozygous individual is showing the same phenotype as homozygous dominant, the type of inheritance is called blank inheritance. Okay, the heterozygous showing the same as homozygous dominant. It means that this one is the complete dominance inheritance. Right, so it's a complete dominant inheritance. If the heterozygous individual showing the blending phenotype of the homozygous and recessive, the type of inheritance is called blank inheritance. So blending is mean that the inheritance is not complete. Okay, so it's gonna be incomplete dominance inheritance. If the heterozygous individual is having both phenotype of the homozygous dominant and recessive, the type of inheritance is called blank inheritance. Okay, like for example, if you combine red and white flower, the heterozygous will be carry or showing both red and white color in the heterozygous individual. So it's called the co-dominant, both of them present. Blank is a family diagram that shows inheritance of a specific disease. So this is called a pedigree. The syndrome that caused by an extra chromosome number 21 is called, this is the Down syndrome. The syndrome that caused by an extra X chromosomes, XXY, is called, this is Klinefelter syndrome. The syndrome that is caused by losing of one X chromosome, like X0, 45X0, is called the Turner syndrome. 191 until 200, this is matching. So you have question on the left and answer on the right. Let's see. 191, homozygous dominant. Of course, it's gonna be with two capital letters, allele. So this is the homozygous dominant. Heterozygous, one capital letter and one lowercase. So it should be this one. Homozygous recessive, it's going to be both carry lowercase allele. Okay. How about the dominant alleles? This should be capital letters, symbolized by capital letter. The recessive alleles will be symbolized with the lowercase letter. Law of segregation. This is the first law, the Mendel first law. Law of independent assortments, that will be the Mendel's second law. Theory of chromosomal inheritance. This will be the Sutton and Boveri. The sex chromosomes, which one is a sex chromosome, will be the chromosome X or Y. Over here we have X, so this is chromosome X. The autosome, example is chromosome number 21. This is the autosome. Okay, so chromosome pair number 1 until number 22, they're all autosome. The rest, X and Y, will be the sex chromosome. Okay, next. Hershey and Chase experiment with bacteria and bacteriophage so that DNA is genetic materials, protein is a genetic material, envelope is the genetic material, spike is the genetic material. So the experiment with bacteria and virus called the bacteriophage. So this virus has capsid, which is protein, and nucleic acid, which is DNA. And according to that result, the DNA is actually the one that enter into the bacteria and duplicate inside the bacterial cell. So this show that DNA is the genetic materials. Watson and Crick models of DNA show that DNA is single-stranded, DNA is double helix, RNA is single-stranded, RNA is double helix. Which one? So Watson and Crick work with DNA, not RNA. So one of these. So according to the models, the DNA is actually double helix molecule. Okay, it has two strands that twisted together. Okay, so it's called the double helix. The monomers of DNA and RNA are monosaccharides, amino acid, fatty acid, or nucleotides. Okay, remember DNA, RNA consists of many nucleotides. So one nucleotide will be look like this. Okay, it has sugar, maybe ribose or deoxyribose, and then it has phosphate, and it has the nitrogen base. Yes, this is one nucleotide. So RNA, DNA consists of many nucleotides. Which of the following nucleotides bases is not present in DNA? Adenine, guanine, thymine, cytosine, or uracil? So we're going to list the base for DNA, which is adenine, thymine, guanine, and cytosine. The RNA base will be adenine. Thymine is not present, so it's going to be uracil, guanine, and cytosine. Okay, look at the question. Which the following nucleotide base is not present in DNA? The one that is not present in DNA is the uracil. Okay, which of the following nucleotide base is not present in RNA? So the one that's not in the RNA will be the thymine. Okay, because thymine only present in the DNA. According to Sargaf rule, in DNA, the number of adenine is equal to the number of guanine, thymine, cytosine, or uracil. So this is the rule for DNA. Okay, A will always equal to thymine. Okay, guanine will always equal or the same percentage with cytosine. And the RNA is similar, but because RNA does not have thymine, so you change thymines with the uracil. Guanine will be with cytosine. Okay, so in the DNA, the number of adenine A will equal to T, so thymine. According to the Chargaff rules, in RNA, the number of adenine will be equal to, so this is adenine in RNA, so A will equal to U, okay, uracil. So that will be the answer. If in human DNA there is 40% adenine, how many percent is guanine? 0%, 10%, 20%, or 40%. Okay, remember adenine is 40%. It means that, okay, there will be 40% of, this is DNA, so it's going to be 40% of time. So 40 plus 40 is already 80. Okay, so the rest, which is 20%, will be for guanine and cytosine. So it's going to be 10%, 10% guanine, and 10% cytosine. So guanine will be 10%. If in human DNA there is 40% adenine, how many percent of thymine? 0, 10, 20, or 40. Of course, thymine, adenine are equal, so there will be 40%. Meselson and Stahl saw that DNA replication is the blank model. Conservative, semi-conservative, or dispersive. Okay, so when DNA separate, okay, the old strands will always combine with the new strand. Okay, like that. So this is what we call the semi-conservative. Which of the following enzyme is used to unwind or break the hydrogen bonds to separate the DNA strands? Helicase, topoisomerase, primase, DNA polymerase 3, or DNA ligase. To break the helix, which is the one that making the helix is hydrogen bond. Okay, so to break these helix molecules, then we need helicase. That's very straightforward. Okay, helicase is the one that break the helix, which is breaking the hydrogen bonds. So when it is broken, then the DNA will be separate like this. Which of the following enzyme is used to glue the hydrogen bonds to join the DNA strands? Glue. Okay. Helicase, this is the one that break. Okay, so the one that's glue the DNA is called the ligase. Which of the following enzyme is used to synthesize the daughter or new DNA strand? Helicase, topoisomerase, primase, DNA polymerase 3, or DNA ligase. So this is synthesis, so it's make polymer, okay, polymerization. Then it's require enzyme that refer the name, which is polymerase. Okay, so this DNA polymerase 3 is the one that adding the nucleotide, making long DNA strand. And this is called a polymerization. Which of the following RNA carrier the genetic code or codon? Which of the following DNA carries the genetic code or codon? So which one that carry the code is called a messenger. This is the messenger RNA. The tRNA is transport. This is the one that carry amino acid. The R is ribosome. This is actually the ribosome that translates the messenger RNA into proteins. How many possible codons are available? There will be 64 type of codon. Okay, and 3 of them stop codons, 1 of them is the start codon. How many types of amino acids are available in nature? There are only 20 type of amino acids. Which of the following codon is the start codon? start codon. Right? So start codon is actually AUG. Okay, August. Okay, AUG. That should be the answer. And the start codons will be using or code for amino acid methionine or methionine. Okay, so this is the amino acid. This is the start, the codon. Which of the following process is not part of proteins translation? Initiation, elongation, termination, or splicing. Okay, remember, translation, it has 3 steps, which is initiation, elongation, and then termination. Splicing is not. Splicing is part of the messenger RNA process. Only cells in the blank will express the gene to make insulin. Stomach, skin, pancreas, or brain. Okay, insulin is made in beta cell in pancreas. So the answer will be cell in pancreas that will make insulin. Only cells in blank will express the genes to make melanin. So melanin is skin dark pigment. Okay, it is made by cell is called melanocyte, and this melanocyte is located in the skin. So the answer will be only cell in the skin produce melanin. (221), 230, there will be true and false. Okay. Nonsense mutation is the type of DNA mutation that has no effects on amino acid sequence of protein synthesis. Nonsense. It means that nothing happened. Okay, so it means that the one that have no effect to amino acid is actually called the silent mutation. There will be no effect on protein that is made. Okay, so the silent mutation. Nonsense will be some amino acid actually will be replaced with different amino acids, causing the proteins will be different. RNA transcription occurs in the nucleus. Yeah, that's correct. Next one, region of DNA upstream of coding sequence is called the promoter. Region of DNA upstream or above the coding sequence is called the promoter. That should be true. If the proto-oncogene mutates into oncogene, it could stop cell division or causing apoptosis or cell suicide. True or false? It should be false because it will not stop. It actually will stimulate the cell division uncontrollably. So it should be false. It causes uncontrolled cell division, causing cancer or tumor. The technique used to amplify specific sequence of DNA is called the gel electrophoresis. True or false? It should be false. To amplify we need what we call the PCR. This is false. It should be polymerase chain reaction or PCR to amplify sequence of DNA. Reproductive cloning aims to produce specific molecules such as hormones and enzyme to cure disease. It is not. Okay, so reproductive is the one that will produce individual clone, is cloning the individual, which is copy of an organism, cloning of an organism. Reproductive cloning aims to produce specific organ, tissue, or cell to cure disease. Again, it is not organ, it is organism or individual, the whole individual. So, does reproductive cloning make copy of organism? Genomics is the study of entire genome, including the complete set of gene, nucleotide sequences, and interaction within species and other species. True or false? Yes, this is true. This is the study of genomics. Norton blotting is the method used to separate DNA sequence. Okay, true or false? It is false. It is not Norton blotting. It is Salton blotting. And for the Norton blotting, it's actually to separate RNA. So you have Norton and Salton. So Salton is for DNA, Norton for RNA. Protein synthesis occurs in the nucleus. Of course, it is wrong. It should be in the cytoplasm because ribosomes are located in cytoplasm. Okay, 231 to 240. This is fill in the blank. You have to write the answer. 231. Blank is the monomer of proteins. What is the monomer of proteins? It will be amino acids. Blank is the monomer of nucleic acid like DNA and RNA. It should be nucleotide. Blank is the organelle that synthesizes proteins. We just mentioned that it is located in the cytoplasm. It's called the ribosomes. Blank is one example of the 3 stop codon. Okay, stop codon, there are 3 of them. Okay, always start with the U. So UAA, UAG, and UGA. UAA, UAG, or UGA. So you just write one of them. The anticodon or amino acid is carried by blank during the process of proteins translation. So which one carry amino acids? This is the T or transport RNA, tRNA. Blank is small circular DNA of bacteria that is mainly used in biotechnology to carry foreign gene. That small circular DNA of bacteria is called plasmid, and plasmid is used to carry foreign gene. Blank is the organism that is modified and receive the recombinant DNA. That's going to be the GMO, genetically modified organism. Blank is a type of cloning that aims to produce identical copy of an organism. This is the one that is reproductive cloning. The 2 strands or double helix of DNA are held together by blank bonds between the opposing nitrogenous bases. What type of bond? This is the weakest type of bond, which is the hydrogen bond. Blank is an example of disease that alters gene expression, causing the cell to divide continuously or uncontrollably. So this is cancer. So cancer is example of disease that alter the gene expression. Okay, so this is the last one. 241 to 250. This is matching. You have a question on the left and answer on the right. Group of about 100 protein that regulate gene expression during transcription. So this should be not 100. I'm sorry, it should be 1,000. Okay, this group of 1,000 proteins that regulate gene expression during transcription is called the transcription factor. And they usually combine at TATA box. Okay. A series of thymine and adenine as part of the promoter during transcription. It is called I just mentioned that that's the TATA box because thymine, adenine. Okay, so this is the TATA box over here. Cell that produce dark skin pigment. It's going to be the dark skin pigment is melanin. Okay, so the cell that produce melanin will be melanocyte. Cell that produce insulin. Okay, insulin produced by the beta cell of pancreas. Stop codon. Okay, there are 3 of them. I just put one, so look for one of them. Start with U. Okay, stop codon start with U. So this should be this one. How about the start codon? This is again August, AUG. Break and wins the double helix. So this is the proteins that break the double helix will be helicase. Join the double helix, the one that glue together, is called a ligase. Repairs the mismatched bases. Okay, if the DNA replication is not correct, if the base is not correct, it will be corrected by this enzyme. The enzyme is called the endonuclease. Cut the specific DNA sequence. This is usually used during the DNA combinations, right? Biotechnology. The enzyme is called the restriction enzyme. It is actually similar with the endonuclease. I think that's all. I hope you will get the best score in your final exam. Goodbye.